All right, our next speaker is Dr. Zhixin Zhang. So let me introduce Dr. Zhixin Zhang. So Dr. Zhang obtains his PhD degree in 2018 from Tokyo University of Marine Science Technology, Japan. Now, he works as a postdoc researcher in Arctic Research Center, Hokkaido University. His research interests include biological innovations, climate change, and conservation biology. In his study, he mainly used species distribution model or ecological niche model to explore species habitat suitability on the different climate scenarios. The fun fact, actually, he said he doesn't have any fun fact to share with us. So let's skip that and let's welcome Dr. Zhang to give his talk.
Okay, Dr. Zhang, thanks for your introduction. So I already recorded my video, so I would appreciate if you can play the video. Then I will answer some questions, if any. Good afternoon, everyone. It is my great honor to present our result in this meeting. I'm Zhixin Zhang, a postdoc from Hokkaido University. The title of my presentation is The Past, Present Day, and Future Geographical Distribution of Seaweed Pipefish. So before the presentation, I would like to make a brief self-introduction. I have broad research interest, including biogeography, climate change, and invention biology. Besides, I'm also interested in conservation biology and crustacean biology. In my research, I use multiple research methods, including modeling, laboratory experiments, and field sampling. And as we all know, the global biodiversity is declining rapidly. And according to the latest IPBES report, there are many indirect and direct drivers that are responsible for global biodiversity loss. And among them, climate change is regarded as one of the direct drivers. Climate change is important because it can influence species in many different ways. For instance, it can influence the sex ratio of certain species. Some marine fish have temperature-dependent sex determination, and ocean warming may result in XX males. And also climate change can influence the species phenology. Some marine fish may spawn earlier in response to climate change. And climate change can also result in novel species interaction. For instance, a study conducted in Eastern Australia, and they found during the past 10 years, due to ocean warming, fish feeding on kelp increase in this region. And therefore, this results in loss of kelp in this region. More importantly, climate change can influence the species distributional range. In response to climate change, terrestrial species can move to higher latitudes and higher elevation regions. And marine species may move to higher latitudes or deeper waters. Therefore, in an era of changing climates, we should understand the species distributional change to better protect, utilize, and manage them. In this regard, species distribution model, SDM, can do this task. SDM can estimate species habitat suitability by determining statistical relationships between species distribution data and the environmental predictors. From this definition, we can see there are three critical elements in SDM. They are distributional data, predictors, and algorithm. So what can SDM do? So in this recent review, authors summarize the applications of SDM. And we can see up to now, SDM has been successfully applied in many different fields. For instance, SDM can be used to evaluate the invasion risk of invasive alien species. They can also use to explore the potential impacts of climate change on species distribution. So now I would like to briefly introduce my target species, the seaweed pipefish. The right two figures shows the seaweed pipefish. This species has fraternal carrier eggs. and it is widely distributed in northwestern Pacific Ocean, including the coastal China, Japan, and Korea. So the recent genetic studies suggest interspecific genetic diversity along Chinese coastal waters with seaweed pipefish. So the Chinese researchers collected seaweed pipefish samples from Yellow Sea, East China Sea, and the South China Sea. And from the molecular result, they found 3 genetic lineage. I'm not familiar with the genetic study. So I'm interested in how to explain this result. So we know the Yangtze River is the largest river in China. And every year, a huge amount of fresh water pour into the ocean. So we call this, and this may serve as a physical barrier for genetic connectivity between Yellow Sea and East Chinese Sea populations. But how to explain the genetic diversity between South China Sea and the East China Sea populations? Is there any historical habitat segregation between the 2 populations? And how might seaweed pipefish respond to future climate change? This is our 2 questions. To answer these 2 questions, we develop SDMs to handcast historical distribution and forecast its future distribution. As I mentioned previously, SDM has 3 critical elements. So I will explain each element in detail. So the first important element is about the species distribution data. So we can collect species distribution data from different online repositories like GBIF and OBRS. Besides, Chinese researchers also conducted a field survey and provided some distribution data. We also collect the data from published literature. So we initially collected 149 records. Not all the records can be used in the model. We should clean the data and avoid clustered record. So this right figure shows the final data used to develop the model. We finally used the 131 records to develop the model. The second important element is about the environmental predictors. We use the different predictors for the 2 models. So for the Palo and the present SDM, we download the predictors from MerSpec. The MerSpec contains present day and the Palo ocean climates. So as for the historical ocean climates, we consider the last glacier maximum. So it is about 21,000 years ago. And as for the present future SDM, we download the ocean climates from by Oracle. This database contains present day and the future ocean climates. And we consider 2 future time periods, the 2050 and the 2100, and RCP scenarios. So the MerSpec and the by Oracle, they contain different predictors. And based on collinearity, we finally select the different predictors for the 2 SDMs. And this slide summarizes the predictors used for the 2 SDM. And the third important element is about the algorithm. Up to now, there are many different algorithms for SDM. For example, there are some regression algorithms, such as generalized linear model, generalized additive model. And there are some machine learning algorithms like random forest and maximum entropy. Actually, different researchers have different opinions about SDM algorithms. Some researchers think each algorithm has its limitations, and we should consider multiple algorithms when developing the model. But some researchers think we should train the parameters for each algorithms. If we find the optimal parameter, the single algorithm can produce a reliable result. So I don't want to see which one is better, but to reduce model uncertainties, in this study we consider ensemble algorithms. We combine predictions with different algorithms. So when developing the SDM, in addition to presence record, we also need the absence data. but the absence data is always difficult to obtain. Therefore, we randomly generated 10,000 points as pseudo absence. And we develop SDM using the biomodel2R package. In this package, there are 10 algorithms, including random forest. But recently, I realized that a random forest cannot handle imbalanced data because we only have 131 record, the absence, the presence record, and we have 10,000 pseudo absence. So the data is imbalanced. Random forest cannot handle the imbalanced data. Therefore, we exclude random forest from our analysis. We evaluate the model performance via 5-fold cross validation. So in short, 80% of the data were used for model development, and the rest 20% were used for model test. And we evaluate model performance using the TSS and AUC values. And we transform the continuous prediction into binary using the 10% presence probability using this threshold. Here, I want to mention that SDM can tell you many information. For example, which predictor is more important and the response curve of each predictor. But in this presentation, I only show you the most important result, the spatial prediction. So first, let's see the payload present SDM result. The right figure shows the model performance. And from the figure, we can see 8 algorithms have a good performance. Therefore, we develop ensemble models using these 8 algorithms. And this figure shows the SDM predictions. The left figure shows the binary result during present-day conditions. The red region means the suitable habitat for the seaweed pipefish. And we can see the large coastal regions of China, Japan, and Korea are predicted to be suitable for seaweed pipefish. And the right figure shows the suitable regions for seaweed pipefish during last glacier maximum. And interestingly, we can see during last glacier maximum, there is a historical barrier between South China Sea and the East China Sea populations. And this habitat segregation may result in genetic differentiation between South China Sea and the East China Sea populations. So this result may partially support our initial hypothesis. So let's see the present and the future SDM. Similarly, this result shows the model performance. And we can see 7 algorithms has good performance. Therefore, we develop an ensemble SDM using the 7 algorithms. And we want to know how will seaweed pipefish respond to climate change. So will they expand their range or contract their range? To answer this question, we estimate the range size change via this formula. So the positive values means range expansion. It means the seaweed pipefish will expand its range. And the negative values means range contraction. So this table shows the result. And we can see generally in the future, seaweed pipefish may expand its range. For example, we can see in 2050, seaweed pipefish may expand about 10% of its suitable range. So to show the results in a straightforward way, I show you this result. The left figure shows the binary result during the present-day conditions. And we can see a large proportion coastal regions of China, Japan, and Korea are predicted to be suitable for the seaweed pipefish. And the right figure shows the potential distribution in the future. So we use the RCP 8.5 in 2050 as an example. And in this figure, the green region means stable. It means this region will be suitable for this species and under present day and future conditions. And the red region means the loss. It means this region will become unsuitable in the future. And the purple region means gain. It means this region will become suitable in the future. And we can see the South China Sea will become unsuitable for seaweed pipefish in the future, but the Yellow Sea and the North Japan will become suitable for this species. So it means the seaweed pipefish may experience northward range shift in the future. So now I make a brief summary. The historical habitat segregation between South China Sea and the East China Sea may partially account for intraspecific genetic diversity of seaweed pipefish. Besides, the seaweed pipefish may experience range shift, northward range shift in the future. And instead of a one-size-fits-all policy, we should consider different management strategies for seaweed pipefish in different regions. So I think this result may have implications for other species in this region. And of course, this study has some limitations and future studies are required. Here, again, I want to emphasize that SDM can tell you more information. For example, the variable importance, the response curves. And the previous study suggests seaweed pipefish prefer certain habitats such as seagrass. In this study, we didn't consider the habitat type into the model. We only consider the marine environment predictors. So in the future, we should consider more predictors into the model. And besides, my recent study suggests lineage level model might lead to more realistic predictions. So therefore, instead of species level model, we should also consider lineage level models for the seaweed pipefish. That's all. Thanks for your kind attention. If you have any question or comment, so please. Thank you.
Okay. Thank you, Dr. Zhang, for the talk. And we can have like a few questions before we take a coffee break, which I should apologize because we should take that like 20 minutes ago. I just missed out. Okay. Any questions? Come on, guys, don't be shy. All right. How about audience online? Are there any questions for Dr. Zhang? Please raise your hands in the room if you have a question. Okay, I can ask the question first. We can take another question if other people are interested. So talking about the predictive map for the Pacific pipefish, the seaweed pipefish, as you show on your slide, there seems like a very big gap in the northeast in Ojai Sea. So the present distribution, there's no present distribution in that area, but you project that in the future, it will expand to this particular zone. So what's the unique feature or characteristic of the environment in this area?
Okay, thanks for your question. Yes, so from the prediction, we can see in the present situation, so the Ojai Sea might be unsuitable for the sea pipefish, but in the future, this region will become suitable for the seaweed pipefish. So instead of answering the question directly, so I don't want to explain why this issue, which predictor is responsible for this phenomenon, but I want to talk about other things. So I think maybe the habitat type in this region is unique. So maybe in the future, the marine environmental predictor, for example, the temperature, might become suitable for the species. So, but the habitat type, maybe there is no suitable habitat type for the seaweed pipefish in the Ojai Sea. So, so my answer didn't answer your question directly. So my point is, in the future, so we should consider the seaweed, the habitat type into the model. So like your previous study about the seahorse. Yeah.
Okay, so I think it's very important to not only get the result about like, where there might be expansion or distribution, but also understand what's the unique features of those different habitats that currently that was not occupied by the species that will be occupied in the future. And I think that ecological meaning would be very important as well. That's one of the comment on that. And another issue is that because this species seems like highly correlated with the seaweed in the region. So have you used any like seaweed habitat data in the model?
Yeah. So this is a good point. So actually, we didn't consider this issue in the model. So when we develop the model, actually I have two strategies to try to consider the seaweed pipefish. So the first is to develop the SDM for the seagrass directly, and then include the SDM prediction for the seagrass into the seaweed pipefish SDM. So this is one of the options, but I don't like this option because the SDM predictions for the seagrass is something like the predictors of the marine environmental layers. So I think maybe in the future we can develop the SDM for the seagrass first, and the binary, and make the seagrass prediction into binary, and we only consider the SDM prediction for the seaweed pipefish only within the suitable range of seagrass. So in short, in this model, I didn't consider seagrass, but in the future, this is one of the optional.